Welcome to RNA and Protein Synthesis, Grade 12, Paper 2. Cells turn DNA instructions into proteins. By the end you'll read translation diagrams, build messenger RNA, and assemble amino acid chains. Get the sequence of steps right and genetics starts to feel familiar. We start small, build a single RNA triplet from a DNA template. The rule here never changes, RNA never carries the base thymine. So wherever the DNA shows an adenine, the RNA writes uracil instead. And cytosine and guanine are always partners, in both directions. Take the three bases, swap each for its RNA partner, and you've built a complementary triplet, the smallest unit of the code. Now the real task, build the messenger RNA beneath that DNA template. Read it left to right and write each base's partner in underneath. Keep one rule at the front of your mind, where the template shows thymine, the messenger RNA uses uracil. That single swap is the most tested idea in this whole topic, and learners lose a mark over it every year. Place each base, glance back at the template, build across the whole strand, and the finished messenger RNA should be complementary and single-stranded. Now watch it resolve in the scratch pad. Each label drops onto its structure. The ribosome wraps the strand. Codons sit on the mRNA. Anticodons sit on the tRNA. The amino acid rides on top. 6 features, 6 correct labels. That's the translation diagram, complete. Now for the journey, and where things actually go. The DNA stays locked inside the nucleus, the cell's master copy never leaves. Transcription builds the messenger RNA right there in the nucleus, using the DNA as its pattern. Then the messenger RNA carries the coded message out through a pore in the nuclear membrane, travels across the cell, and reaches a ribosome. That is where translation happens, in the cytoplasm. Nucleus, out the pore, to a ribosome. Watch the journey play out. The DNA stays put in the nucleus. The mRNA forms there, then carries the coded message out through the nuclear pore. It reaches the ribosome in the cytoplasm. The tRNA stands ready with its amino acid. Every molecule is exactly where it belongs. Translation happens at the ribosome, the large structure wrapped around the strand. The bases are read in groups of 3. On the messenger RNA, a 3-base group is a codon. On the transfer RNA, the matching group is the anticodon. That is the most common label swap on the diagram. Easy anchor, the codon sits on the message, the anticodon sits on the taxi that delivers each amino acid. Here is game 02 resolving. Each RNA base drops into place beneath the DNA template. A becomes U, T becomes A, C swaps with G, watch the mRNA grow, U, C, A, then G, G, C, then A, C, 8, 9 positions, all correct. The model is biologically complete. Now match each codon to its anticodon. The codon sits on the messenger RNA, and the anticodon is its mirror on the transfer RNA. They must fit like a key in a lock. Use the same pairing rule we built earlier, adenine pairs with uracil, cytosine pairs with guanine. Read each codon, write the complementary anticodon beneath it, then dock the transfer RNA into place so it delivers the next amino acid. Here are the 3 codons, and the tRNA carriers drop in beneath them. UCA pairs with AGU. GGC pairs with CCG. ACA pairs with UGU. A pairs with U, C pairs with G, and each amino acid rides in on its carrier. All three docked. The match is complete. Now we build a chain from the DNA triplets. Read them left to right, like a sentence, one triplet at a time. Use the code table to turn each triplet into its amino acid, then place that amino acid in its socket and the chain grows in order. Exam tip: the table is usually written for messenger RNA codons, so make sure you work from the messenger strand, or you'll drag the wrong amino acid. Link them together with peptide bonds. The chain builds itself now. AGT gives serine. CCG gives glycine. TGT gives threonine. Each amino acid snaps into its socket, in order. Then the peptide bonds form between them. Serine, glycine, threonine, linked into a protein chain. Final challenge, and this is the gift question. For biological errors are hiding in this pathway, and each carries a mark. First, an RNA strand shows a thymine, but RNA never uses thymine, it should be a uracil. Next, a triplet on the transfer RNA is labeled a codon, on transfer RNA it is the anticodon. Then the amino acid link is called a hydrogen bond, proteins join with peptide bonds. And translation is in the nucleus, it happens at a ribosome in the cytoplasm. Correct all four, then order the propositions, transcription first. This is the full repair, and you watch every fix land. The base becomes U, not T. The tRNA triplet becomes an anticodon. The amino acid link becomes a peptide bond. Translation moves to the ribosome in the cytoplasm. Then the 12 propositions secure in order, from the DNA unwinding to the finished protein. Every idea in place. The model is complete. You've now walked the whole route from DNA to protein. Remember the spine: DNA stays in the nucleus, messenger RNA carries the message to a ribosome, transfer RNA brings the amino acids, and peptide bonds join them into a chain. Hold on to that picture and a surprising number of genetics questions resolve themselves. Practice the route, it comes back every sitting.